Hello everyone. In last class we discussed about Ramachandran plot and protein folding and unfolding and also the protein unfolding diseases. So the main diseases related to the misfolding of protein. In today's class we will be discussing about methods of protein structure prediction. Before going to the methods of protein structure, we should know why we need to learn about protein structure prediction. So as we discussed in the first class, the protein structure is determined or classified into various categories, that is primary structure, secondary structure, tertiary structure and quaternary structure. Whereas the functionality of the protein or the functional protein will be formed only in the tertiary or quaternary structure. So the function of the protein or functionality of the protein has many processes. So the main processes are catalysis, cell movement, cell signaling, cell transportation, cell regulation. These are some of the functions of the protein which is involved in the human body, not only in the human body, and also various other organisms. And what is the significance of studying protein structure prediction? So we are able to predict the sequence of the protein and also be able to predict the structure of the protein from the sequences, and also be able to predict the function of the protein from the structure and also from the sequences. So these are the main significances of the studying protein structure prediction. As we know, the biological processes of all human body or all living organism is mainly depends on the structure and function of the protein, and also be able to detect the evolutionary relationship between the proteins of various kinds or from various species, and also be able to predict the structure of the protein, 3D structure of the protein mainly, because 3D structure of the protein is the main cause of the functional protein, and also be able to detect the misfolding diseases which is related to the protein structure. And also protein structure determination help us to find the novel proteins, and also the protein structure prediction mainly involved in drug development and drug discovery, since the functionality of the protein is also very important in the formation of the particular drug. There are two main ways in determining the protein structure. In the first case, the protein is unknown. So in that case, protein has to be extracted from the source and then it has to be purified. After that, the purified protein has to be separated based on the chains, and then the fragmented chain has to be sequenced. Once it is sequenced, the reassembling of the fragments is based on the overlapping fragments. This is how the first case will go on. Whereas in the second case, the protein sequence is known. We can say the protein sequences has been already present in the databases. In that case, again we have to retrieve the protein from the source and then purify the protein and then separate chains and fragment the protein and then sequence them. Whereas the reassembling of the sequence is mainly based on the sequences which are present in the databases which are already submitted in the protein databases. Now we are going to see the challenges which are influencing protein sequence prediction. So from the experimental point of view, we have to know the challenges involved in the protein sequence predictions are: it is very laborious, time consuming, and then it is very cost effective. And also when we solve the protein structure using methods like crystallography and Nmr spectroscopy, it needs skilled persons, and also it has to be trained in a proper way to get the proper structure of the protein. And also the cost of the novel protein prediction, or to get the sequence of the novel protein, it will be around $1 lakh to $10 lakh. These are the challenges we are facing in the experimental methods, and also with even today's new technology, we will not be able to predict the structure as soon as possible or as early as possible. These are some of the problems or challenges we are facing in determining the protein structure. There are 4 main levels in protein structure analysis. The levels are atomic scale method, and then binary interaction method. The third one is complex interaction method, and finally cellular method. So what is atomic scale method? The interaction between atoms and residues will be determined in this method. So the major methods or experiments used for this analysis are X-ray crystallography and Nmr spectroscopy. And the next one is binary interaction method. So in this method, the interaction between two proteins will be assessed, and also the chemical interaction cannot be assessed in this method. This is the major disadvantages in the binary interaction method. The third method is complex interaction method. That is, the multiple proteins can tend to form a complex. So the complexity or the interaction between each proteins will be assessed in the complex interaction method, and also the chemical interaction cannot be determined in this method too. And then finally the last method is cellular method. So the proteins will be involved or will be interacted with the cellular components like cell membrane, plasma membrane, or organelles like nucleoplasm or nucleus or cytoplasm. So the interaction between the cellular components and the proteins can be assessed in this method. These are the four main methods in protein analysis. So now we are going to see about the experimental methods which are used to determine the protein structure prediction. There are various methods with there to determine the protein structure prediction, but now we are going to see only few methods. We are categorizing the methods into three main levels. That is, first one is sequencing method, second one is secondary structure prediction method, and the third one is tertiary and quaternary structure prediction method. So in the sequencing method, we will be discussing about Edman degradation method and mass spectrometry. Whereas in the second method, that is secondary structure prediction method, we will be discussing about circular dichromism and then Fourier transform infrared spectroscopy, that is FTIR. And in the final method, that is Quaternary and tertiary structure prediction method, we will be discussing about X-ray crystallography, NMR spectroscopy, and Cryo electron microscopy. So now we are going to see about the sequencing method. So under sequencing method, we will be discussing two methods. One is Edman degradation method, another one is mass spectrometry. So in Edman degradation method, the proteins will be purified by removing one residues at a time at the N terminal. And also Edman added one chemical called phenyl thioisocyanate because the proteins tend to damage because of the hydrolytic condition. So by using that chemical, it will create a derivative called phenyl thiocarbamyl derivative. This derivative which helps the protein not to be degraded in the hydrolytic condition. And the next method is mass spectrometry. As we know, this mass spectrometry is an analytical tool where in this method, the molecules will be assessed or determined based on the mass by charge ratio. So the whatever molecules or components present in the sample will be determined based on its molecular weight. So with the help of the molecular weight, we will be able to determine each protein structure in the mass spectrometry. Now we will be discussing about secondary structure protein prediction, under which we will be discussing about two main methods, that is circular dichromism and FTIR. So circular dichromism is a rapid method for determining the secondary structure of the protein, and also we will be get to know the protein folding conformation also. And in circular dichromism method, we will be able to analyze the protein expression and protein folding, protein unfolding, and also if the mutation occurs in the protein, and we will able to get to know the structure of the protein if the mutation occurs. And also the stability of the protein can also be considered or determined using this method. And also we will able to determine about the protein-protein interaction by this method. Another method is FTIR. So the full form of the FTIR is Fourier transform infrared spectroscopy. As the name indicates, this method will occur at the IR range. So the secondary structure of the protein could be obtained at the amide regions at the ratio of or a range of 1600 to 1700 frequency. So by this frequency, we are able to get the amide groups present in the protein. And also we know the proteins will have a different spectral band at different regions. So based on the spectral band regions, we will get to know the protein structure in detail with the help of the FTIR. Till now we saw about sequencing method and secondary structure protein prediction method. Now we will be discussing about tertiary and quaternary structure prediction methods. So under this category, we will be seeing about three main methods, that is NMR spectroscopy, X-ray crystallography, and finally Cryo electron microscopy. So now we will be seeing about NMR spectroscopy. What is NMR spectroscopy? It is mainly depends on the nuclear spin, and this nuclear spin will help us to generate the magnetic field. So when the magnetic field is applied, this nuclear spin will be oriented either in the favor of the magnetic field or against the magnetic field. And also when the magnetic field is not applied, the nuclear spin will be randomly in direction inside the instrument or inside the process. So based on the nuclear spin orientation, we will be get to know the structure of the protein. And this NMR spectroscopy help us to determine the kinetic reactions and also protein structure analysis at the atomic level. So next method is X-ray crystallography. So by this method, we are able to assess both the atomic and molecular level of the protein. So the principle behind X-ray crystallography is X-ray diffraction pattern. So when the crystalline protein or the crystalline atom gets the X-ray, it will give away the X-ray beams in different specific regions. Based on the diffraction pattern, we will get to know about the protein structure. In this method also, we will get to know about the atomic levels or atomic interaction between various biological processes. The last method under this category is cryo-electron microscopy. As the name indicates, the process will occur at the cryogenic temperature by using electron microscopy. Under this method, the protein interactions and also the cellular structures and also virus complexes, protein complexes can be determined at the molecular level. So in this method, the samples will be in the native state and also we don't need to use any dyes to get to know about the interaction. Till now, we saw about the various experimental methods to determine the protein structure. Now we'll be discussing about the disadvantages of those methods. When we look at the X-ray crystallography, so in X-ray crystallography, the protein has to get the well-ordered crystal structure. If you're not getting the well-ordered crystal structure, then we'll not get the final protein structure. It will not be in a good conformation. And when we look at the Nmr spectroscopy, the protein has to be in a soluble condition for Nmr spectroscopy. And also, the protein size has to be very limited for Nmr spectroscopy. And mostly these experimental methods are very expensive and time consuming also. And also, the very biggest problem in determining the protein structure is how we are going to relate the amino acid sequences with the 3D structure of the protein. This is the major problem in experimental methods. Now we are going to see about the computational methods needed for studying about the protein structure prediction. So under this category, we will be seeing about two main levels. So first one is the methods for secondary structure and the second one is the methods for tertiary structure. Under secondary structure, we will be discussing about three main methods, that is Chou-Fasman method, GOR method and neural network method. Under tertiary structure prediction method, we will be discussing about three main methods, that is homology modeling, ab initio method and threading method. Even though there are so many methods under computational methods, we will be discussing about these methods only. So the first computational method to determine the secondary structure of the protein is Chou-Fasman method. This method is widely used method to predict the protein structure. And also this method is mainly depends on the propensity value of the protein. So propensity value is the tendency of the amino acid residues present as an alpha helix or beta sheet conformation is called propensity value. So we can calculate the propensity value based on the frequency of the amino acid present in the alpha helix by frequency of the total residues present in the protein, by which this way we can predict the propensity value. And also the propensity value is mainly depends on the R group present in the protein. R group plays a crucial role in determining the propensity value based on which the secondary structure of the protein can be determined. The next method is GOR method. This method is an information theory based method. It has been developed in the late 1970s after Chou-Fasman method. It is based on the concept of sliding window concept. And the final method is neural network method. This neural network method is based on the concept of neuronal cells. That is, the signaling of the neuronal cells will be passed from first neuron to another neuron. Like this way, neural network method is generated. So the neural network method has three layers, that is first layer, second layer and then hidden layer. So the output of the first layer will be passed as an input to the second layer and the information will be shared between the hidden layer, so that the hidden layer will be present in between the first and the second layer. This is how the neural network method will be performed. Now we are going to see about the computational methods for tertiary structure prediction. We have three main methods, that is homology modeling, threading and ab initio method. So the first two methods, that is homology modeling and threading, is mainly depends on the sequence databases. That is, the target protein will be related with the sequence databases. For example, if your target protein is similar to the database sequence for about 40%, then homology modeling is preferred. For example, if your target protein is similar to the database sequence below 40%, then fold recognition model is preferred. So homology modeling will construct and give you the atomic model of your protein, whereas this fold recognition method, your target protein will model as a fold protein or fold structure and compared with the fold structures that are present in the database sequences or databases structures. And then finally, we have another method is called ab initio method, which is entirely different from these two methods. So ab initio method does not need any sequence databases to be compared, whereas this method will generate a model from scratch, and it's mainly based on the physical properties rather than the solved structures. These are the three main methods to predict the tertiary structure of the protein, and this is how the structure of the protein will be predicted computationally. We came to the end of the session today. Hope you all understand about methods of protein structure prediction. We saw about various methods related to the experimental and computational methods to determine the protein structure, and in next session we'll be dealing with the X-ray crystallography. Thank you all.